Yeah. I waived the need to sign in. So. Okay, I think we're going to get started. And my role, my name is Charles Hart. I'm the director of the Catalyst program here at UCSF, which is UCSF's Translational Accelerator. And my role today is just to welcome you all to UCSF and to introduce Michael Hostetler, who's a chemist, but also an attorney at Wilson Sonsini. So, Michael. Great, thank you, Charles. So, thanks everyone for coming to this award ceremony for the third drug hunter. You know, I think it's particularly relevant that we're holding this in Genentech Hall, in part because of Richard, as you'll see, his role in creating Rituxan, which probably led to a lot of profits for Genentech, and maybe some of what he's done has helped build this hall today. So it's sort of fitting that he is getting the award ceremony and the award presentation in Genentech Hall today. I want to also thank the four entities that came together to create the drug hunter. The idea of a drug hunter is really someone who has, I would say, a good nose for a drug. And that's part of it. But then you also really need the grit and the drive and the energy to bring that drug to market, because that takes a lot of work. It's one thing to discover it. It's another to drive it forward to commercialization. And I think what we recognized is that Richard is one of those rare individuals that really combines the nose for a drug as well as for someone who has really the grit and energy to drive it forward. So I think it's very fitting. And he's also, I think, one of the few individuals that has actually brought both an antibody and a small molecule to market. That's actually pretty amazing if you think about that too, because there's people that bring antibodies to market and other people that bring small molecules to market. Really, Richard is probably one of the handful that has done that for both. And on top of that, they're both billion-dollar selling drugs. So that's pretty cool also in that regard. As Richard will go through, Rituxan was, I think, uniquely so, one of the first major biologics to get approved. And then not only get approved, but to have really exceptional sales. And I think in some ways you could credit that for the ability for many other biologics companies to come in and say, hey, I can do that too, and follow on. So again, I think Richard's role in all that, we wanted to recognize as part of this. And really, if you think about that, probably millions of people have taken these drugs. And so a lot of lives were changed for the positive. A little bit about Richard. He'll talk a little bit about his life, but right now he is currently CEO of Corvus Pharmaceuticals. And on top of that, he is still a practicing oncologist. So he still sees patients. He sees it through his clinic is it Stanford? He's got a clinic at Stanford that he still sees patients. He still has residents that he works with and trainees. So on top of being CEO, he also does that. He also has the interesting background that he was Chief Commercialization Officer at the University of Texas. So he was familiar with the concept of university-industry relationships, and I think that's another key thing that is part of his DNA. The other thing I want to mention, which is almost mind-boggling, in his career with the startup companies that he's had, he has raised over $1 billion in investments. I mean, think about that, $1 billion in investments from the companies that he started with, going all the way back from CellPro, IDEC, Pharmacyclics, Principia, and Corvus, $1 billion. So really unusual in that regard. And then on top of that, I just want to call out Richard for still publishing in the scientific literature. He's got a recent article in Cancer Discovery, just out in 2020, where he was a senior author. So he really is driving this forward. So all told, there's really a great pleasure that we award Richard Miller as the third drug hunter. The first one was Steve Caldor, who has brought a number of diabetes drugs to market. Rich Heyman was the second drug hunter who helped bring a number of oncology drugs to market, including those for treating breast and prostate cancer. And Richard has done an amazing amount of work
bringing drugs for treating a variety of lymphomas, as you'll see. In addition to the plaque, which we will be presenting to Richard, we're also donating $1,000 to the charity or the worthy cause of his choice, and it's going to be the Stanford Cancer Center. So we're very happy to be doing that, and we're also, it's going to be really cool to hear about how Richard helped bring both Rituxan and Imbruvica to market. So with great pleasure, let's welcome Richard.
Thank you. Oh, thank you. It's not quite the Super Bowl, but it's pretty good. Thank you, Mike, for those remarkably generous words. I hadn't realized I was that old or did all those things, but I had a lot of help, as you'll see. I want to first of all thank the UCSF Catalyst Program, the University of California Drug Discovery Consortium, the Center for Discovery Innovation, the Catalyst Program, and then finally, of course, Wilson Sonsini. So what I would like to do today is, I prepared a talk the other day, and I was presenting it to one of the UCSF faculty, talking about it, and then I started telling him about some of the challenges and sort of the things you don't really think about when you start companies and you do this. And he said, Richard, that's what you ought to talk about. Forget all the science. So I'm not going to show a lot of blots and DNAs and things like that, but I did want to sort of give you a feel for the history of how these things happen. Because especially when I deal with students, and I deal with students and residents and fellows, as Mike mentioned, in my clinic, sometimes they have no perspective for like the way you used to do things, and they just sort of take things for granted. So what I want to try to do is sort of go through some of this history. So let's start with the first slide. The key message that I want to leave is that if you focus on one disease and you study it for a long time and really carefully, you can be pretty good at it. And I've had the, I guess, good fortune to study one disease really, really carefully, read all about it, see patients with it, and just really be totally immersed in it for over 30 years now. And so that has given me, perspective and advantage in doing this kind of work. So when I was finishing my fellowship at Stanford, monoclonal antibodies, the Kohler-Milstein technique had just been published a few years earlier. And being very naive, I thought, well, antibodies are going to be the greatest thing going. And this was a cartoon. I think it appeared around 1980 in New Yorker magazine. And I used this a lot because I thought this is the way the world would be, that antibodies were going to be used to cure everything. And what you see here is a picture of a doctor answering the phone saying, call me in the morning, take some antibodies and call me in the morning. Of course, this is incredibly out of date for many reasons. First of all, antibodies are great drugs now, great agents, and they have taken over many areas of medicine. But it's not quite like that. And it took a really long time to produce these antibodies, as you'll see. You'll also notice doctors don't really talk to patients like that anymore. It's usually done through email, through a secure email, through the hospital. They also don't wear ties and white coats like that. So this is remarkably out of date. But the fact is that we all thought that antibodies were going to cure the world. And here's what happened. This is the timeline. It's pretty sobering, actually. And I think the historical perspective is useful. So Kohler and Milstein paper in Nature 1975. That's an incredible paper. It's only like 2 pages. Kohler and Milstein won the Nobel Prize in 1984 for that paper. And what they did was they immunized a mouse with sheep red blood cells. And then Milstein, who was studying genetics and through cell hybridization, looking where chromosomes would go. They had the clever idea of, well, if we do that with B cells, maybe, and we drug mark them, maybe we can immortalize them and come up with monospecific, monotypic, or monoclonal antibodies. And they did. And they showed in that paper that they had a monoclonal anti-sheep red blood cell antibody. And it just grew and grew and grew and produced gobs and gobs of antibody, and this was fabulous. And they hypothesized that you could do that with other other antigens as well. It did not take long for people to notice the importance of that article. Everybody jumped on that immediately. One of my mentors, Len Herzenberg, who discovered the flow cytometer, happened to be on a sabbatical in Milstein's lab in Cambridge, saw that technique, stopped his sabbatical, ran back to Stanford, did the same thing, and people were making monoclonal antibodies to all kinds of things very, very quickly. It did not take long. And the thing about this technique, it was so robust, it worked. You would go in the lab, and if you followed the recipe, it would work, and you'd come up with these antibodies. So that's why I was naive enough to believe that cartoon, that, boy, we're going to make antibodies to cancer and infectious diseases and viruses, and we're going to cure the world. So the first antibody approved for therapy didn't happen for 10 years. It was an agent called OKT3, Johnson & Johnson, or Ortho. OKT3 was a mouse monoclonal antibody that was approved for treating kidney rejection. If you got a kidney and you were rejecting it, you got prednisone. If that didn't work, then you get OKT3. It was extremely toxic, as you can imagine. It's a mouse antibody. It had cytokine release syndrome and all that stuff you hear about now. And it didn't do very well in the marketplace. Now, this was not a crazy idea, because at the time, people were using horse antithymocyte-globulin and rabbit antithymocyte-globulin to treat rejection. So this was thought to be a big improvement over that. But still, it didn't do well in the marketplace. And then it took another almost 10 years. And Barry Kohler at Mount Sinai, Stony Brook, and then Mount Sinai, then Rockefeller, very good scientist. Barry Kohler made a human antibody to what's called glycoprotein 3 on platelets, and that was an anticoagulant. It was a Fab fragment. It got approved by the FDA. And he had the actually very clever idea of using it with stent placement. So if you were going to get a stent, and everybody got this, and stents were big around this time, if you were going to get a stent, you got an injection of this Fab fragment before the stent, and then for about 12 hours after. And that was standard practice for a long time. I believe it was marketed by Lilly, but Lilly didn't make the antibodies produced by somebody else. Anyway, that wasn't really a therapeutic. And I would argue this wasn't really a therapeutic either. But then in 1997, another few years, Rituxan was approved in November of 1997. And Rituxan was really, really a breakthrough, because it was very effective, it was very safe, and it was very versatile, as you can see, as I will show you. Now, very shortly after Rituxan, about a year later, Herceptin was approved. So Rituxan, Herceptin, suddenly you had big drugs. I mean, everybody was using this stuff. Everybody with B-cell lymphomas. Herceptin, of course, was only useful for the 15 or 20% of breast cancer patients that are HER2 positive. But these two agents, particularly Rituxan, showed the pharmaceutical industry that these drugs could be big, big money makers, and people would use them, and they're practical, and they're easy to use, because before this, pharmaceutical executives were very, very worried about, how do you do this antibody stuff? I remember I remember Ron Levy and I going up to see the CEO of Bristol Myers on Park Avenue in New York about lymphoma antibodies. And I was sitting down talking to him, and he said to me, so these are proteins, right? I said, well, yeah, they're antibodies. They're kind of proteins, yes. And he said, do you have to refrigerate those? I said, yeah, that would probably be a pretty good idea. He said, well, we don't have refrigerators in our oncology clinics. And Bristol Myers at the time, you're talking about the time when there was Cytoxan and cisplatin and etoposide and bleomycin and stuff. This was the biggest oncology cancer company in the world by far. And he was worried about the fact that they didn't have refrigerators in the oncology pharmacy, and they didn't. Now, here I was trying to figure out, how do you produce this? How do you make it? How do you screen for these antibodies? How do you purify it? And this guy's worried about a stinking refrigerator. And so I was, freaking out over this. But anyway, fast forwarding now, this really showed the world that you could make monies, and these were going to be practical, safe, effective drugs for the pharmaceutical industry. And now, if you go 20 years later, roughly, there's over 100 monoclonal antibodies that are approved for therapeutic purposes. Obviously, there's a lot of other antibodies that are approved for diagnostic purposes, but the diagnostic part of it is easy. I mean, those antibodies are very easy to make. Now, there's a couple of things to realize or to think about. So what was the world like before Kohler and Milstein's paper in 1975? Well, I'll tell you what the world was like. First of all, the idea of using antibodies for therapy was not a new idea. I mentioned there was horse antithymocyclobulin and rabbit antithymocyclobulin. There was actually anaserum that was, I've never seen it used, but apparently it has been used. Antibodies to digoxin were used to treat digitalis poisoning. Digoxin is a drug we don't use anymore, but it was a very good cardiology drug. Many people took it, and it had a very narrow therapeutic index. So if it got if you had toxicity, because it had a very long half-life, you had to get rid of it. I've never seen that antibody given. There were, of course, antibodies, as you know, for rabies, to neutralize rabies and snake bites and things like that. But really, the most successful antibody, which was an anaserum, is still used in your obstetrics department, and it's called Rogam. And it is actually marketed by Ortho. So Rogam, in an Rh-negative mother who has an Rh-positive baby, so about 10 to 15%, I believe, of women are Rh-negative, and if they have an Rh-positive baby, they can make antibodies to Rh, and the babies get what's called hydrops fetalis. That's a very bad disease, fatal. And so Rogam is still used, and it's basically plasma from immunized, I think they get it from immunized men. But before Kohler and Milstein, if you wanted to make antibodies, you had to go immunize an animal, you had to go bleed the animal, you had to purify the antibody, and of course it wasn't an unlimited supply. Whether you used a rabbit or a goat, it was only so much antibody, and you'd run out, and then you'd have to go do it again, and of course it would be different. And so this offered the opportunity to get a continuous supply of monoselective, monospecific drug. That made it possible to have a drug. So I would say the beginning of antibodies for lymphoma, or actually for therapy, because nobody had done this yet, was this paper in the New England Journal of Medicine in 1982. And this was an experiment. I made fun of it this morning. This was N=1 trial. N=1 trials are really good if you really study these patients carefully and take every blood sample you can and every specimen you can get from them, which is what we did. And so the idea here was we were fixated, as the scientific community was, on finding tumor-specific antibodies, because you're always afraid that the antibodies would react with normal antigens and perhaps be dangerous and toxic. So it turned out that George Stevenson, a scientist in the UK, had described the fact that the immunoglobulin on a B-cell lymphoma was actually specific for that tumor. It's monoclonal. And that variable region is called an idiotype. And so you can make anti-idiotypes. People who study this are just called idiots. Because it's only going to be good for that one patient. But we did that. We spent months making an antibody to this patient's tumor that was an anti-idiotype. Now this was really hard. I mean, making the anti-idiotype was, or finding it using hybridoma technology was not hard, but producing the antibody in large quantities was hard. The first thing that we did is we grew up a ton of the hybridoma in cell culture and purified it, got maybe a couple hundred grams, and tested it. I remember running back and forth between clinic and the lab doing a rabbit endotoxin test. And that, because in those days you didn't really have these fancy ELISAs for endotoxin. And so the typical test was you inject a rabbit and you see if they get a fever. And typically they get a fever at about 2.5 to 3 hours. And so this antibody that we had worked months and months on, we injected in the rabbit, and I was running down the clinic and reporting to my people, oh, well, gee, the rabbit's great, it looks good, it's doing great. And then at around 2.5 hours, this poor thing was looking really pretty bad. Hyperemic, febrile, and it was back to the drawing board. And we learned that if you made it as an ascites, you can get much more concentrated antibody, and you didn't have any endotoxin, and that's what we actually used. So this was a mouse monoclonal anti-human B-cell idiotype. Now this patient had very advanced follicular lymphoma. He had failed everything. He had a very low platelet count. He had no neutrophils. He had massive adenopathy and splenomegaly. He was a wreck. He came into clinic febrile, and my colleague, Ron Levy, was away, and I had the antibody ready, and I was afraid if we didn't use it, we might lose the opportunity because he was going to die. I remember, you know, in those days you didn't need an IND. If it was in the institution, you could do it if you had IRB approval, and we had that. So I remember going to the chairman of oncology and explaining to him, listen, we got this antibody, the guy's got no other therapies, I want to give him this antibody, and so forth. And he looked at me and said, well, do you think it'll work? And I said, well, I don't know. It binds to his tumor cells. And he said to me, you know, I wouldn't do that. I don't think you should do it. I think you should give him some more Velbam and prednisone. I walked out of the office, his office, and I knew right there and then that I had to do it. It was the right thing to do. Because all he could recommend to me were some drugs that he'd already failed. And we treated him, and he had a complete remission in about a month. His platelet count came up, his fever went away, his lymph nodes shrunk, he felt better, and we ended up publishing it in the New England Journal. That man stayed in complete remission. He was about 72 or so when we treated him. He died not too long ago, actually. He became a good friend of ours because he would always come to clinic, and he was very grateful. He died at, I think, 97 years old from other causes, of course. He never had a recurrence of his lymphoma. So you can see when this story was told, people got pretty jazzed up. This was pretty exciting. There were just a couple of problems here. Number one, this was a mouse monoclonal antibody, and lymphoma patients are very immunosuppressed, so they may not make antibodies to your product. And number two, this was a patient-specific drug, so you'd have to make one for each patient. Nowadays, we're actually doing that, of course, with various gene therapies and so forth. But in 1982, people thought you were crazy. So just to get back to what the world was like then, around 1990, an oncologist named Sandra Horning was in an article in a magazine, and she said she was an authority on non-Hodgkin's lymphoma, NHL. She ran what's called the Eastern Cooperative Oncology Group, which at the time was the best lymphoma cooperative group. And she said, you know, lymphoma therapies hit the wall. We've had a lot of drugs come along in the 70s and 80s, chemotherapy drugs, and we keep putting these chemotherapy drugs together, but we're running into toxicity. You can't just keep putting cytotoxic agents together. You're going to get, obviously, cumulative bone marrow toxicity. And so she said that breakthroughs are going to depend on novel mechanisms of action and tolerability. And she was right. So this was the beginning of a very, Sandra became professor of medicine at Stanford, president of ASCO, hundreds of publications, CMO, chief medical officer of Genentech and Roche for 10 years. And this was the beginning, this really motivated me and was the beginning of a very important collaboration that resulted in me marrying her and having 2 children. So, okay, so let's get back to the evolution of rituximab. No, it's a good collaboration. I mean, Who's got a better collaboration than that? So let's get back to this story. So we published anti-idiotype for follicular lymphoma. We said, let's go, and we did a few more patients, I should say, and the results were interesting. I mean, we had most patients respond. And we said, okay, let's go start a company. And we went to Brooke Byers. This hall is named after Brooke Byers. Brooke Byers was a partner at Kleiner Perkins Caulfield and Byers. And we convinced him to put some money into it, along with some other firms. And so IDEC was started in around 1985. And the idea, the story was that, look, we start with this patient-specific stuff. We're going to learn what we're doing. And then but we're going to find antibodies that react more general to patients' idiotypes. Because we knew about what are called shared idiotypes and so forth, or framework determinants and so forth. And we actually did come up with a panel of antibodies. By 1989, this was also published in the New England Journal, we came up with about 25, 30 antibodies that reacted with 30 or 40% of lymphoma patients. And so the idea was like going to get contact lenses. They measure your eyes and stuff, and then you go in the back room and they pull out the contacts that are ready for you. Or I used to say, like going into a shoe store, you pick out your shoes and they measure your foot and you get the right pair of shoes. Well, there's no free lunch, however, because one of the other things we learned about giving antibodies is sometimes there's antigen in the way. And antigen's a problem. Antigen forms immune complexes is what you give, and you get immune complex disease. So it was nice that these idiotypes were shared by the lymphomas from patient to patient, but they were also shared by the normal circulating immunoglobulin, and so there was just too much of a barrier to get through to get the tumors. So that idea didn't work. But then the next idea was, well, wait a second. What about a more generic antigen? And right around this time, Lee Nadler at Harvard, who I think is the unsung hero in all of this, he discovered CD20. We said, well, I mean, this antigen was really great. It was on B cells, sustained in intense expression on B cells, normal B cells, and most B cell malignancies, 98% of them. No other tissue. So we said, well, okay, well, let's make an antibody that would be more generic, not patient-specific, that you could just use like any other drug. And in fact, the rest is history. That antibody was called C2B8. And that led to a phase 1 study and FDA approval, which I'll show you. Now, interestingly, making antibodies is about screening. The person who actually made that antibody, Sarah Hart, who was a technician working in my lab, now as a teacher, I think she teaches high school in Santa Rosa, California. We just burnt her out, I think, or whatever. But Sarah made this antibody very cleverly. She took a T-cell leukemia from a patient. She got some of the B cells from that patient. She transformed them with EBV. So she had a B and a T cell from the same patient. She immunized with the B cell and screened on the T cell. Obviously they're HLA identical, so a lot of the other antigens would be eliminated in the screening. And we were able to lickety-split come up with an anti-CD20. Now other people were making anti-CD20s too. This was not 20s as well. So this wasn't that unique. But the big breakthrough was just a few years before that. Greg Winter in England, Cambridge, published a paper on engineered antibodies, making chimeric or engineered antibodies. Greg Winter won the Nobel Prize in 2015 for doing this. And so progress was slow because we could get away with giving mouse antibodies to lymphoma patients because they're so immunosuppressed. But in solid tumor patients, they're not as immunosuppressed, and they would very quickly make antibodies to the antibody you gave. So the bioengineered recombinant antibodies were a big breakthrough, and we were fortunate to hire a guy, Mitch Reff, who did that with the B-cell antibody that Sarah Hart made. He was able to make that chimeric. And then it went into a phase 1 trial and then phase 3 trial, as I'll show you. So this is Rituxan schematically. Rituxan is an IgG1 kappa, fully capable of fixing complement, binding to Fc receptors. It has a mouse variable region, though. So the first 100 amino acids or so of the heavy chain and light chain are mouse. The rest is human. Turns out I have never heard of a single patient, to my knowledge, no patient has made anti-mouse from getting Rituxan that I'm aware of. I've never seen that reported. And I calculated the other day, and this is conservative, there's probably well over 5 million people who've gotten Rituxan. And that's not counting arthritis or anything like that. So it's pretty straightforward. Now these days, of course, we can do better. We can engineer the framework so they're also human as well. But I'm not sure it mattered. But the key to Rituxan, as Dr. Horning mentioned needing, was the safety and versatility, novel mechanism of action. I would say to this day, we don't really know how Rituxan works. I don't know what the mechanism of action is. But you can find papers showing that it induces apoptosis of lymphoma lines in vitro. You can find papers showing that it will fix complement and mediate complement cytotoxicity. You can find ADCC and natural killer cell mediated effects. You can even find papers that show T cell involvement in some cross-antigen presentation. But I would say we don't really know. But what we do know is that even patients who have great responses and they relapse or become resistant, they always have CD20 on their cell. So we never see a mutation or a selection for a CD20 negative cell, strangely enough. And by the way, I don't know if anybody knows what the function of CD20 is. I think that's also a matter. I don't know what the function of CD20 is. So this was the phase 1 trial. This was the first time I'd ever seen what we now call a waterfall plot. And a waterfall plot is that tumor reduction is down, so down is good, negative tumor size, and growth, of course, is positive. And this was about 115 patients with relapsed late-stage follicular lymphoma. And as you can see, most of these patients benefit. So the argument to the FDA was, hey, listen, I give this patient, and most of them, maybe they don't all get complete responses, but they're all benefiting from a treatment that's pretty easy and pretty safe to give. And that was a powerful story. I wish I could get waterfall plots like that more. Now that led to the next study. This was a classic, very important paper by Coffier in France through GILA. GILA was a cooperative group in France. They could pump out studies really quickly. This was an aggressive lymphoma called diffuse large B-cell lymphoma. The other lymphomas were slower growing, follicular lymphomas. This is an aggressive lymphoma. And this article published in the New England Journal showed a survival advantage when you added Rituxan to what was called CHOP. Now CHOP is a chemotherapy regimen that's 4 drugs, Cytoxan, Adriamycin, vincristine, and prednisone. It cured about 40% of patients. These days you add Rituxan to that and maybe the cure rate's up to 60%. So that was a pretty big change. So it got approved for first-line large cell lymphoma. And then, of course, you know the paradigm. You get drugs approved late line and then you move them up early. And that's what happened here. This was a study done by ECOG in follicular lymphoma, first-line therapy. And you don't see curves like this very often, PFS curves. The standard therapy for follicular lymphoma was Cytoxan, vincristine, prednisone, CVP. Add Rituxan to that, and you improve the progression-free survival from 1.3 years, it goes to 5.1 years. With a p-value, we've got a bunch of zeros, 7 or 8, and you don't need, again, a statistician to figure out that that's highly significant. So these days, we don't even give the CVP. So if you have uncomplicated follicular lymphoma, you're not too sick, you'll get Rituxan alone, and that'll work in most patients. Now eventually they relapse and need other things, et cetera. But drop the chemotherapy. That's a big deal, because that chemotherapy, vincristine neuropathy, the side effects of prednisone, Cytoxan makes you lose your hair, and I don't like that. So Rituxan's approved for a lot of things. It's approved for all kinds of different lymphomas and Waldenstrom's macroglobulinemia and all kinds of things like that. But it's also approved for autoimmune diseases, rheumatoid arthritis. It's approved for psoriasis. I think it recently got approved for pemphigus. And the autoimmune part of this was really something that was unanticipated because everybody was thinking T cells, T cells are going to be the way you suppress people and prevent autoimmune disease. But it turned out that, and there were, during this period of time, everybody was giving anti-T cell antibodies to patients with autoimmune disease, and that was not working. It was not working, and it was dangerous, and it was toxic. And eliminating B cells or depleting B cells was working. How does that work in autoimmune disease? I don't know. Does it block antibody formation? Nobody's ever shown that. Does it block antigen presentation? I don't know. But it does work in a lot of different autoimmune diseases. So here's where the rubber meets the road. This is the most important slide. The rest of it is meaningless. This is a slide taken recently from the SEER report. That's the epidemiology branch of NIH. That looks at lymphoma, non-Hodgkin's lymphoma, of which most are B cells. New cases, of course, from 1980 to 2020 were rising and sort of hit a plateau. If you look at the death rate from lymphoma, you see the curve bend in 1997, the year Rituxan was approved. There is nothing else that, I mean, this slide has been shown to many, many authorities. There is nothing else that can explain the bend of that curve but Rituxan. It's not antibiotics, it's not any of that stuff, it's got to be Rituxan. And you can see that the 5-year survival, which is shown in these gray boxes, went from 50% up to over 70%. So that really made a big, Rituxan has made a big, big difference in lymphoma management. The fellows at Stanford call, because we use it all over the place, they call it vitamin R. And I think that's a good term. Okay, well, Rituxan paved the way. There's been second, third generation things. There's biosimilars now. Rituxan now, there's a form of Rituxan called Hycela, which can be given subcutaneously. Patients love that. It's really a great advance. Highly recommended if you need it because you don't have to get an infusion. Gazyva is a new and improved Rituxan that has a better ADCC function. Not being used that much yet, but I think it actually is better than Rituxan, although this is debatable. In a randomized trial, it was a little bit better, but because there was no survival advantage, people don't use it. I think those survival curves will separate with more time because this is a very long disease, and if you look at the original data, it looks like those curves are just starting to separate. It is more toxic. It causes more infusional reactions. And then I think the other big one is Ocrevus. So ocrelizumab is also anti-CD20. Again, a different carbohydrate and ADCC function. Ocrelizumab has completely changed multiple sclerosis. So Rituxan we knew worked in multiple sclerosis. There was an article in the New England Journal a long time ago. Actually, I think it was published by Steve Hauser here at UCSF, showing that MS had a lot of MS patients, multiple sclerosis patients, had a lot of B cells in their brain, often oligoclonal. And in fact, he did a trial with Rituxan and showed improvement in MRI. And then it was just sort of dropped. Nobody actually ever pursued it because there were other treatments coming along for MS. Ocrelizumab is the fastest product launch in the history of Roche. It has become the dominant treatment now for multiple sclerosis. It's one injection every 6 months. Patients like that. And it seems to have very little toxicity. So again, it's a B-cell depletion play. Okay, but here's the problem. No matter how good you are, the next morning you wake up, people say, okay, like what can you do for me tomorrow? And IDEC and Biogen had a problem. IDEC had Rituxan. Biogen had Integralin for MS, but there was really nothing else. And when you're enjoying market caps that they had, people are saying, okay, well, what's the next rodeo? And that led to a merger, basically equal merger where the two companies merged, doubled the market cap, and then eventually Biogen dropped the name IDEC. but that led to that. And at the time, a $6.8 billion deal in 2003 was a pretty big biotech deal, maybe one of the largest. Before I leave Rituxan, I want to talk about something I think Mike mentioned, that I used to run licensing commercialization office at UT for 1 year. I did that for 1 year because when I left Pharmacyclics, I have some very good friends in the chemistry department at UT, and they said, Richard, you got to come here and help us commercialize stuff. So I told them I would do it for 1 year, and I did. I've always been very interested in the academic industry collaboration and how we should be better at exploiting that and utilizing it. And IDEC and Stanford represented the very best of that. First of all, the lab and Dr. Levy's laboratory at Stanford were 15 minutes away. Technicians would be driving back and forth. I would be driving back and forth. Patient samples would be transferred. Reagents that we would make would go to his investigators. There were publications produced. And it was just a really good collaboration that resulted in a lot of progress and a good drug. And I fear that we've destroyed that with conflict of interest policies and all kinds of other things that I realize do have some importance, but I'm afraid that we maybe have overdone that, and we've now put up some impediments to the exchange, and I think that's slowing things down a lot. But anyway, the reason I like this picture is because with this picture, since the picture of me is much later, I can say that Ron was my student because he looks much younger. But he wasn't my student. So now I want to talk a little bit about ibrutinib. Now, ibrutinib is, in some sense, a much easier story because the kinases were already known. So you were just riding down a road that was already paved, but except for one thing that I'll tell you about. So I left IDEC, went to Pharmacyclics, and we were able to get our hands on a drug that we called PCI 32765. That sounds like the number of a prisoner in Les Miserables. And again, I was thinking B cells, lymphoma, because that's the only thing I knew. I never did anything. I don't know how to play golf or anything like that. So everything was B cells and lymphoma. But this one was really easy because I didn't need to do any experiments because Mother Nature had already done the key experiment. And that key experiment was described by Dr. Ogden Bruton in 1952. Most of the people here weren't born in 1952. I was 1 year old. So I didn't really know him, but he was a pediatrician in the public health service, and he described a disease in boys, who had no antibodies, no immunoglobulin in their serum, and really no B cells. They had defective B cell development. And that was a pretty cool experiment. Of course, now we know that this disease that Bruton described, which is now called Bruton's agammaglobulinemia, is due to a mutation in what's called BTK. And BTK has a very strategic location near the B cell receptor, the surface immunoglobulin, and signaling antigen binds the B cell receptor, signals through BTK, and makes B cells divide and differentiate and make antibody and stuff like that. So we said, okay, well, Rituxan does good stuff. Maybe if we interfere with B cell function in this manner, we could have a role in lymphoma. And in fact, it amazed me that nobody had thought about that before. And when I asked about that, people said, oh, well, there's this paper that says if you block BTK, it won't work. And that's always a good tip-off. When people say that, go read the paper. Because when I went and read the paper that everybody was referring to, it had nothing to do with B-cell lymphoma. It had, it was something about pre-B-cell ALL. It was like crazy. It didn't mean anything. So in any event, we developed a drug, a kinase inhibitor for BTK. And this is nothing smart here. This is, I mean, like many other kinase inhibitors, it had your purine backbone and a bunch of stuff hanging off of it. But it did have one thing that was different, and that is this group right here, which is an acrylamide. And an acrylamide is reactive. It's an electrophile. It seeks a nucleophile. such as like sulfur. And cysteines, of course, have sulfur. Well, people had made compounds like this as tool compounds to react in the laboratory, but they never thought about using them clinically. And there was a good reason for that, because everybody would refer to these two papers. Sometimes publishing papers are really bad. And these I would say were bad. They're actually good papers, but what these two papers showed is that they made correlations between reactive intermediates, acrylamides in particular, and idiosyncratic drug reactions, forming adducts. If you form adducts indiscriminately with proteins, you can do bad things like allergic reactions and things like that. And there is some truth to that, but we weren't trying to make protein adducts indiscriminately. We had a drug that bound into the pocket of the enzyme. And as shown here, so this is ibrutinib binding into LCK. There was no crystal structure of BTK at the time. LCK was close, so that seemed to work. And the key here is that the electrophile would react with this cysteine. Now there are about 11 cysteines that have, 11 kinases, known as the TEC family, that have a cysteine in the same position, which is remarkable. I've always thought that's really remarkable. Why is it that they all, even if you look at dogs and mouse and everything, they all have a cysteine in that position? What is so, and it's a free sulfhydryl. Why is that? And I don't think the answer to that is known. But anyway, we tried for months playing with the molecule, making non-covalent versions of this molecule. And everything we did, we found that the molecule with the electrophile worked better. And finally, one day we said, well, wait a second. Why are we trying to get rid of the electrophile? The patients we want to treat have cancer, and we're giving them cyclophosphamide and cisplatin and adriamycin and things that form adducts with their DNA. Who cares about forming some adducts with their protein? And by the way, we're going to do it very specifically. And in fact, so we started to develop this as a drug. And I won't go through the science here, but basically the remarkable thing about the covalent drug was that you had these really cool assays you could do where you would take B cells, you could incubate them with the drug, you could wash them. Now if you had a regular reversible drug and you wash the cells, the drug is gone, it doesn't work anymore. But because this was covalent, you could wash these things all night long and you still maintain their inhibition of B cell proliferation or activation as was measured by flow cytometry. So some really cool assays. So basically crystal structures and functional assays show that this would bind like Velcro, it wouldn't come off. And because it had to be in exactly the right position to react with that cysteine, it conferred an extra degree of specificity, selectivity. So rather than being a negative, it was actually improved your selectivity, and because it was so potent and so durable mechanism of action, it actually improved your therapeutic margin. It was safer. And I won't go through this. Basically, you could show the same thing, showing that it blocked the downstream signaling of BTK by looking at phospho-PLC gamma, ERK, etc. We don't need to go through that. And it worked in autoimmune disease remarkably well. So here's your typical collagen-induced arthritis. So if you do this, your diseased animals are up here in the black. They get arthritis, an arthritis score. If you give them Decadron, which is always the positive control, they don't get it. And if you gave them 32765, you could prevent it. If you stop treatment, it comes back. You could even treat established disease. If you waited until day 8 when they had florid arthritis, you actually could get reversal of established disease. That's really remarkable. But lymphoma, we weren't interested in arthritis. We're more interested in lymphoma. And the mouse models just didn't look very effective. They're transplanted mouse tumors, and it was really hard to convince ourselves that we were doing anything good. So we turned to man's best friend, and that is dogs. So dogs get lymphoma. They get B-cell lymphomas and T-cell lymphomas. They get a B-cell lymphoma that looks very much like human disease. And in collaboration with workers at Colorado State University, in one of our first publications on this drug, we found that, in fact, dogs responded. They had tumor regression. Now, dogs may be man's best friend, but they're not man's best patient. And it's a little hard to work with them. They don't cooperate. They don't give you good history. They don't like sitting in the CT scanners. PET scans are really a bitch in them. So we said we got to get into patients here. So there are problems with dogs, but it did give us the first clue that this could be effective in people. And in fact, Ranjan Advani at Stanford published this paper in a phase 1 trial. Now you've seen the waterfalls. We've switched the direction of them. But you can see that patients with refractory relapsed B-cell lymphomas and a variety of histologies, CLL, mantle cell, DLBCL, et cetera, were responding to this treatment, and it was very well tolerated. So that was the phase 1 trial. And of course, the pharmacokinetics confirmed what I told you in the laboratory, which is that if you took the blood, you could take the blood out of the patients, look at their B cells, and you could show that even though the drug was clearing from the plasma quickly, in a matter of hours, there was persistent inhibition of the BTK. So it was occupied above 90% for 24 hours when the drug is gone. So the pharmaceutical guys were going nuts on this because they couldn't figure this out. You know, like they're trying to do AUC curves and stuff like that, and this is not like, you know, we said, no, forget that stuff. So I like this drug because I didn't really need to, because I never did well in that part of medical school. And so this way I didn't even really have to think about that. It's all about getting the drug in there quickly, and getting out quickly is actually good. So we call this a PK, pharmacokinetic pharmacodynamic disconnect. This led to a phase 3 trial by John Bird in CLL, relapsed CLL. You can see the curve. I mean, that's a staggering PFS curve. You don't need to be a statistician for that one either. And there was also a slight survival advantage. And this is now one of the most widely used drugs for CLL, mantle cell lymphoma, and a couple of other things, Waldenstrom's, macroglobulinemia. So again, that question comes up. Hard enough to develop one drug, how do you develop a pipeline? And this led to AbbVie purchasing Pharmacyclics for one of the largest deals I'm aware of in biotech for $21 billion. Now that's even a more staggering number because Pharmacyclics had licensed 50% of the rights to J&J, so you can multiply that by 2 in terms of the value. Okay, because time is short, I'm just going to tell you that at my company Corvus, I have to say something about Corvus. We're interested in a kinase called ITK. It's the homolog of BTK, but it's in T cells. So why are we interested in this? Well, there's T-cell lymphomas, there's autoimmune disease, and there's a lot of other things that we'll learn along the way. We've been able to make a very selective ITK inhibitor. This is the dendrogram. Look at sunitinib, that's Sutent. Approved, it's a kinase inhibitor approved for renal cell cancer and a few other things. Ibrutinib was way more selective. Look at this one, only hits one protein. And if you put this in mice with autoimmune disease and lymphoma, it's kind of a mouse model that they get lymphoma, but they get autoimmune disease too. You can cause reduction of their lymphadenopathy, improvement in their kidney function, and this led to a dog study, same playbook. And this is Chloe and Rudy. Chloe had what's called an aggressive or peripheral T-cell lymphoma, and she had, I think it's a she, I'm not sure, had a reduction of lymphadenopathy. Rudy had a reduction of a cutaneous T-cell lymphoma involves the skin in dogs around the snout, and people all over the body. And you can see these dogs have regression of involvement of their skin. And this drug has recently gone into a phase 1 trial, and we presented this data last week. And this is a PET scan from a patient with who's had what we call a nodal CR, cutaneous T-cell lymphoma. This is a PET CT scan. There's activity there, and that's in the right hilum, this is in the subcarinal area, and it's basically gone. So we're sort of following the same playbook. Now I wanted to talk a little bit about the future because I've been through this game before. You know, what you did yesterday doesn't count. And so I think we're in a really difficult spot now in general in the field because there's so much going on. And the pace of research and the pace of discovery is faster than we can do the clinical trials. And I think one of the challenges now is, like, if a clinical trial takes 5 years to do or 3 years to do, like, is anybody going to care when you're done with it? And so, like, this is a real challenge. So you already know what's going on, bispecific antibodies and targeted therapies and checkpoint inhibitors and cellular therapies, CAR T, et cetera. And how do we keep up with all this? Well, one of the ways we have to respond to this is by developing better, more innovative, more flexible clinical trial designs. And fortunately, that has been happening, and the FDA has been very good about accepting some of these newer techniques. Adaptive trial designs. We write one protocol with a lot of what-if scenarios, and you can just expand expand groups, cohorts of patients, and answer many questions. And we've been doing a lot of that, and it really allows you to move faster. Novel endpoints, employing biomarkers that help you understand that your drug is doing what you intended to do and have some relationship to the disease. Statistical analysis. I just used to think that, hey, stats is, like p-value, okay, I get it. But no, there's newer statistical techniques now that allow you to compensate for the multiplicity of analyses and do all these sorts of things. So I would say the key takeaways for me is very simple. Know the disease. If you want to develop a drug for a disease, you better really know that disease. And that doesn't mean a 5-minute conversation with the guy down the hall. That means really knowing the disease. Secondly, I think you have to know the patients. And I think you have to take those initial few patients and study them really carefully. Biopsy everything you can get. Get blood as often as you can get, urine, get whatever you get. Spinal fluid, I don't care. Get whatever you can because you will find things that you didn't anticipate. And then finally, know what dogma to ignore. I was thrown out of every drug company because we had a drug with a reactive intermediate. And once they saw that, which I didn't tell them until the end, they would walk out of the room, say like, oh, what are you kidding me? So that's not very good. Or a guy who tells you, hey, we don't have refrigerators in the oncology clinic. So how are you going to give an antibody to patients? So know what to ignore. And that's a line I stole from an article about Francis Crick, Watson Crick, the aspirin, a reporter asked Francis Crick, what does it take to win a Nobel Prize? Well, what's your reason for success? And he says, well, I was just really good at knowing what to ignore. And I think there's a lot of truth to that. So I want to end by, again, thanking the sponsors and everyone for inviting me to this delightful lecture. I want to thank a lot of people at IDEC. Obviously, one person doesn't make a drug. It takes a lot of people with all different sorts of skills. At Stanford, I want to pay a particular tribute to Ron Levy, my mentor, Henry Kaplan, my mentor, Saul Rosenberg, my mentor. I had the privilege of training with the best lymphoma people in the world at the time, and others in the oncology division at Pharmacyclics, Joe Buggy, Eric Ben, David Lowry, many others. Many of those have transplanted to Corvus, and of course, all the patients in the hospitals and the collaborations that we had were really fantastic. So I want to thank everyone for coming and paying attention to this boring, sobering story.
Okay, let's congratulate Richard on a fascinating career and well-deserved 3rd Annual Drug Hunter Award. And there's a reception right now hosted by Wilson Sonsini that you're all welcome right out these doors. So thank him one more time.
Thank you. Thank you. Thanks, Mike.
That was great.
Thank you. I hope that
That was really good.